The Spectrum Top 50 method is developed and conducted by Roma Labs, covering over 50 different mycotoxin and metabolites. For example, the main mycotoxins, aflatoxins, zearalenone, A and B trichothecenes, and so on, as well as frequently occurring mycotoxins, masked mycotoxins, emerging mycotoxins, and other secondary metabolites. What are masked mycotoxins actually, and why are they an emerging issue for feed and food safety? So the fungus in the plant produce mycotoxins, for example deoxynivalenol. And then a defense mechanism of the plant is going into place, so the plant is adding sugar or any other substance to the mycotoxins, so the mycotoxin is masked. For example, DON3 glucoside is built. And then, after the animal ingests contaminated feed containing masked mycotoxins, this sugar is cleaved in the gut again and the parental mycotoxin is released. So the harmful DON is then available in the gut again. And up to 45% of the total DON in a cereal sample can be present as DON3 glucoside, and these masked mycotoxins cannot be detected by conventional analytical methods. Here I will show you an overview about the results of all the samples that were analyzed with the Spectrum Top 50 method in 2024. In total, you see that 10,096 samples were analyzed, and there on the left side you have a kind of color code. So each color represents some kind of mycotoxins. For example, the orange one leads to regulated or guidelines mycotoxins. For example, the first prominent one is deoxynivalenol. It occurs in 74% of all analyzed samples, with a median value of 350 ppb and a maximum of 21,000 ppb. And you see a very prominent color is the blue one, which is colored for emerging mycotoxins. So we see here bovarecin and eniatine also occurs in almost 70% of the samples. Another available method is the Spectrum 380 method. This is a research method conducted at the university, detecting more than 800 different mycotoxins and metabolites, as well as bacterial and plant toxins and metabolites. And here, in this figure, there is an overview about how many metabolites are found per sample. And we see that almost half of the samples contain between 20 and 39 different mycotoxins, and the average number of all mycotoxins and metabolites per sample was 39. Thank you for joining me for this short video. To find out more about different methods in detecting mycotoxins and metabolites, please explore also the other videos.